The cell is often thought of as the fundamental unit of life. Viruses are of course non-cellular entities, but many scientists do not consider viruses to be alive. There is enormous diversity in cell structure, especially among microorganisms. The specific size, shape, and organization of microbial cells are determined by both the physiochemical and ecological conditions faced by different microorganisms. The lives of microorganisms are governed by physical and biological principles that are fundamentally different from those governing macroscopic organisms. Physicochemical factors that affect cell size include rate of diffusion, surface to volume ratio, and viscous forces. Biological factors that affect cell size include predation and parasitism. At the microscale, movement and mixing of nutrients and other essential compounds are governed by passive diffusion rather than turbulence, which is the norm at the macroscale. The rate of diffusion therefore sets limits to how fast a compound can reach a cell from the outside environment or move from one part of the cell interior to another. For example, at 20 degrees centigrade, it takes a molecule of glucose 1.1 millisecond to move 1 micrometer by diffusion through water. However, a 10-fold increase in the distance to 10 micrometers results in a 100-fold increase in the time required, 110 milliseconds. At a distance of 1 millimeter, the time required for the glucose molecule to travel this far is 19 minutes. The rate of growth for any microorganism is ultimately determined by the rate at which the cell can acquire nutrients and dispose of waste products with its surrounding environment. The rate of exchange between the cell and its surrounding medium is in turn governed by the amount of available surface area for transfer to occur. Smaller cells therefore have proportionally more surface area per cell volume where transfer of nutrients and waste products can occur. Movement of a solid object, such as an animal or microbial cell, through a fluid medium is governed by forces of inertia, the momentum of the moving object, and the viscosity of the fluid. In the case of larger objects, such as an animal, the inertial forces dominate so that even if an animal stops swimming, its motion will continue due to its momentum. Microorganisms experience a very different environment where viscous forces dominate. Consequently, the motion of a microbial cell that stops swimming will cease immediately. This property is described by the Reynolds number, which is the ratio between inertial forces and viscous forces and is calculated using the formula shown below. The Reynolds number lacks units, and a high value means that inertial forces will dominate, while a low value means that viscous forces will dominate. An adult human of average height, 1.8 meters, swimming with a speed of 1 meter per second, has a Reynolds number of just under 2 million. Conversely, an Escherichia coli cell measuring 2 micrometers, swimming with a speed of 30 micrometers per second, has a Reynolds number that is more than 10 orders of magnitude lower. The hydrodynamic environment faced by microorganisms is therefore often referred to as a low Reynolds number environment. This diagram shows the remarkable spectrum of organism sizes of viruses, archaea, bacteria, and eukaryotes. Note that the scale is logarithmic. The range of cell sizes among microorganisms clearly shows that there is no single optimal cell size. It is governed by external conditions. The lowest size limit for a cell must at least be large enough to fit its genome and metabolic machinery, including ribosomes, which have a diameter of 20 to 30 nanometers. Obligate intracellular parasites with reduced genomes are therefore also often small in size, such as mycoplasma and chlamydia. The upper size limit for a cell is determined by whether metabolites and proteins can be transported within the cell with sufficient speed, either through passive diffusion or active transport mechanisms. The genome must be capable of sufficient rates of RNA synthesis to satisfy the demand for protein production. Some large eukaryotic microorganisms are multinucleate to keep up with the demand for RNA. Small cell size is the norm among prokaryotes and can be found in some eukaryotic microorganisms as well. The benefits of small cell size include that less biomass must be synthesized prior to cell division, greater surface-to-volume ratio for nutrient uptake from the external environment, passive diffusion is sufficient for intracellular transport of metabolites. The drawbacks of small cell size include greater vulnerability to predation by protists, greater difficulty to move through aqueous media due to a lower Reynolds number. Despite the challenges of larger cell size, some bacteria have adopted cell sizes exceeding 100 micrometers. Epulopiscium fischelsoni is a gut symbiont of the brown surgeonfish and varies in size during the daily rhythm of its host. Epulopiscium repeatedly duplicates its genome as it grows, so that the amount of genomic DNA is proportional to its volume. The largest cells of Epulopiscium can contain hundreds of thousands of copies of its genome, as shown in the graph. Bacteria of the genus Thiomargarita are the largest bacteria described to date, with a diameter of up to 750 micrometers. Thiomargarita bacteria live in sulfide-rich marine sediments where they extract energy by oxidizing sulfide into elemental sulfur by respiration. Rather than using oxygen as an electron acceptor, Thiomargarita bacteria use nitrate, which is more readily available in their oxygen-starved environment. Since nitrate is not always available, Thiomargarita bacteria devote some 98% of their cell volume to storing nitrate. Therefore, the effective volume of a Thiomargarita cell is actually much smaller, just a thin layer between the nitrate storage compartment and the cell membrane. This much smaller volume allows intracellular metabolites to diffuse at rates that are compatible with a typical prokaryotic cell. There is great diversity in cell morphology among microorganisms. The majority of microbial species maintain a distinctive morphology with the help of a cell wall, some form of protein scaffold, or external shell. Conversely, some microbial species do not maintain a distinctive cellular morphology, such as amoebae. As with cell size, the wide diversity of cell morphology among microorganisms demonstrates that there is no single optimal cell shape. Instead, the specific cell morphology of a particular microorganism reflects adaptations to the physico-chemical and ecological conditions of its environment. For example, spiral-shaped cells can move through viscous media, such as mucus, more efficiently than spherical or rod-shaped cells. Cell morphology is an important diagnostic tool in the identification of bacterial species. Bacteria that grow as single cells can typically be assigned to one of the 6 basic morphologies shown below. However, this classification scheme is somewhat subjective. For example, what degree of curvature separates a straight rod from a curved rod, and what threshold separates a spherical cell from an ovoid cell, and so on. Some bacteria that grow as single cells have the ability to change cell morphology in response to environmental conditions. For example, Listeria monocytogenes will transition from a rod morphology to a coccus morphology in response to extended nutrient starvation. If the nutrient-starved cocci are supplied with fresh medium, they will transition back to a rod-shaped cell morphology. Haloquadratum wolsbii is an archaeon found in hypersaline lakes and was the first microorganism with a square shape to be described. The flat square shape of the haloquadratum cell is an adaptation for more efficient harvesting of light energy from the sun. Gas-filled vesicles along the edge of the cell keep the cell suspended in a horizontal orientation to maximize the amount of light that hits the cell. Unicellular microorganisms divide by one of 2 main mechanisms: binary fission or budding. Binary fission produces 2 identical daughter cells, while budding results in a larger mother cell and a smaller daughter cell. The daughter cell produced through budding must then first attain full size before it can itself become a mother cell. Many microorganisms grow as shorter chains or long filaments, hyphae, of cells that do not physically separate after cell division. Some microorganisms alternate between unicellular and filamentous morphologies, so-called dimorphic microorganisms, while others are strictly filamentous. Filamentous growth, also known as hyphal or mycelial growth, confers a number of potential benefits over unicellular growth. Filamentous growth can enable a microorganism to bridge microscopic pockets of low nutrient availability, a very common situation in soil ecosystems. Nutrients are transported from cells in high nutrient environments along the filament to cells in low nutrient environments. Filamentous growth is also a strategy to avoid engulfment by predatory protists by simply becoming too big to be eaten through phagocytosis. Filamentous growth also allows for specialization of individual cells within the filament. One example of such specialization are the nitrogen-fixing cells in filamentous cyanobacteria known as heterocysts. Due to the oxygen sensitivity of the nitrogen fixation process, heterocysts cannot perform photosynthesis and instead receive fixed carbon from neighboring cells while simultaneously supplying these cells with fixed nitrogen. Hyphae in filamentous fungi are divided into three main categories. Pseudohyphae, where individual cells or hyphal compartments are separated by cell wall septa without any interconnection between cytoplasms. Septate true hyphae, where individual cells or compartments are separated by septa, but septal pores enable interconnection between cytoplasms. Aseptate true hyphae, where the hypha forms a single continuous compartment without septa. A mycelial clone begins with either a germinating spore, such as from actinobacteria or fungi, or a single cell switching from unicellular growth to filamentous growth, such as with dimorphic fungi. A germinating spore undergoes isotropic expansion until a polarity axis is established, and an initial germ tube begins to extend along the polarity axis. The germ tube then becomes a primary hypha that continues to be propagated by polar extension at its tip, or apex, where the deposition of new membrane and cell wall occurs. Since growth only occurs at hyphal tips, the microorganism will form hyphal branches to increase the number of hyphal tips so that the surrounding growth substrate can be effectively colonized. Lateral branching occurs some distance from the hyphal tip and establishes a new and separate polarity axis. Apical branching occurs at the hyphal tip, which is typically observed in rapidly growing hyphae. In filamentous fungi, individual hyphal compartments may either be uninucleate or multinucleate, while filamentous actinobacteria, such as Streptomyces species, have largely aseptate hyphae with multiple genome copies. Fungal hyphae are notable for their rapid extension and ability to penetrate solid substrates such as live or dead animal and plant tissues, so-called invasive growth. The force required to achieve both fast hyphal extension and invasive growth is thought to be generated mainly through high intracellular hydrostatic pressure, also known as turgor pressure. The hyphal network essentially functions as a microhydraulic network. Since the cell interior typically has a much higher solute concentration than the surrounding environment, osmotic pressure increases as water will tend towards entering the cell from the outside environment. By allowing water to enter hyphal compartments behind the hyphal tip, this increases hydrostatic pressure inside the hypha. The cell wall prevents the hypha from rupturing, while septa towards the center of the mycelium are closed off. Therefore, the hydrostatic pressure gradient is directed towards the hyphal tip. At the hyphal tip, the increased hydrostatic pressure is used to extend the hypha as new membrane and new cell wall material is deposited at the apex. The hydrostatic pressure is also used to drive efflux of water at the apex. Many cellular microorganisms, especially those with a predominantly unicellular lifestyle, possess physical appendages that project from the cell envelope. Such appendages can consist of flexible filaments, often made from protein, or rigid spikes or spines of silica or calcium carbonate. Some of these external appendages are highly specialized for particular functions, while others can be adapted for several uses. Functions of external cell appendages include locomotion, surface attachment, communication between cells, avoiding predators, and predation of other microorganisms. Locomotive appendages can be found in all three domains of cellular life: archaea in domain archaea, flagella in domain bacteria, cilia and flagella in domain eukaryota. Eukaryotic cilia and flagella are functionally identical, and the term undulipodium is sometimes used to encompass both structures. The word flagellum is used to describe long undulipodia, which are often present in numbers below 10, while the word cilium is used for shorter undulipodia that are typically present in higher numbers, hundreds or thousands. This diagram shows the cross-section of an archaeolum, a bacterial flagellum, and a eukaryotic undulipodium. Note the relative simplicity of the prokaryotic structures compared to the eukaryotic undulipodium. Archaea and bacterial flagella consist of thin protein filaments that are rotated by a basal motor complex to generate torque for locomotion. The archaea motor is powered by ATP hydrolysis, while the flagella motor is powered by a chemoosmotic gradient of either protons or sodium ions from the cell exterior. Archaea and bacterial flagella can be rotated both clockwise and counterclockwise, which results in reverse and forward motion respectively. These images show the diversity in flagella arrangements in bacteria depending on the number of flagella possessed by the bacterial cell. Unlike prokaryotic archaea and flagella, eukaryotic cilia and flagella do not rotate, but instead propel cells through fluid environments through the creation of a whip-like waveform. Eukaryotic cilia and flagella consist of an axoneme that is composed of a bundle of 9 doublet microtubules surrounding a central pair of single microtubules. The entire structure is covered by a cytosolic matrix and a specialized ciliary membrane. Waveforms are created by dynein motors causing microtubules to slide past each other on one side of the axoneme, which causes the entire structure to bend. Dynein motors on opposite sides of the axoneme alternate in causing microtubules to slide past each other in a coordinated fashion to create specific waveforms and beat frequencies suited to the movement of the cell. This diagram summarizes the differences in locomotive appendage function between archaea bacteria and eukaryotes. Note the run and tumble states of peritrichous bacteria. This mode of swimming motility allows bacteria to respond to external stimuli such as concentration gradients and thereby regulate their movements.